Our third speaker, Glenn Werneburg, is a dual MD PhD candidate in the Department of Molecular Genetics and Microbiology, working in Dr. David Tanasi's laboratory. His research focuses on understanding the mechanisms by which bacteria cause disease. Glenn is interested in both basic science and clinical medicine and believes that his formal training in both will allow him to translate scientific discoveries into improvements in diagnostics and treatment of human disease. His talk today is entitled Dissection of Usher-Chaperone-Subunit Interactions during Pilus Biogenesis in Escherichia coli. Escherichia coli. A political scientist. Why am I? Glenn.
Okay. Thank you, Dr. Tabor, for the introduction, for the invitation to speak. I'm Glenn Werneburg. I'm in the Tanasi lab, and I know my title is a bit of a mouthful, but the concepts that underlie my research are pretty easily graspable, so we'll go through them together. In essence, we're interested in how bacteria cause human disease, and we ask that question so that we can prevent these bacteria from causing disease. And the disease that my bacteria cause is urinary tract infection. UTIs are a very, very common infection. Nearly 50% of women will have a UTI in their lifetime. It's also very expensive to treat and diagnose. Over $1 billion go into the treatment and diagnosis of UTIs in the US every single year. Clinically, UTIs manifest as pretty much deviation from any normal urinary pattern. So things like dysuria or pain on urination, increases in urinary frequency or urgency to urinate, hematuria or blood in the urine, fever, flank pain, all of these are clinical indications of UTI. And UTIs are generally diagnosed based on a patient history and physical exam. For example, if somebody has pain on urination and urgency to urinate, there's a 90% chance that a diagnosis of UTI would be correct. These UTIs can go on to cause and progress to sepsis, shock, and death in some cases. Actually, this is exactly the sequelae from which Pope John Paul II died in 2005. Treatments for UTIs are generally antibiotics, and depending on whether the UTI is present in the bladder, or in the case of cystitis, or in the kidneys, pyelonephritis, we use different first-line antibiotics. These antibiotics are becoming less and less effective due to the phenomenon of bacterial resistance, and that underscores the need to develop new classes of these antibiotics. So the idea is we want to understand how these bacteria cause infection so we can identify these novel alternatives to the traditionally used antibiotics. How do UTIs come into being? The human urethra is normally a sterile place, and the urethra can become contaminated with fecal flora. Fecal flora contain many species of bacteria, including uropathogenic strains of E. coli, my model system. And these uropathogenic strains of E. coli, or UPEC, can ascend the urethra and gain access to the human bladder. And mediated by pili, as shown in this electron micrograph, these bacteria can specifically adhere to urinary epithelium on the bladder. Further, other strains can further ascend up the ureters and gain access to the kidney. Again, mediated by pili, they can adhere specifically to the kidney. So these pili project out from the bacteria, they have adhesive properties, and they can specifically interact with cells within human tissue. How are pili assembled? Pili are assembled by a pathway termed the chaperone-Usher pathway. It's a secretion system similar to the one Lawton had mentioned. It's used by gram-negative bacteria to assemble and secrete these adhesive structures onto the surface. And it's not only used by E. coli, it's used by many different bacteria to facilitate their capacity to cause infection. So besides E. coli, things like Klebsiella pneumoniae, the causative agent of pneumonia, particularly in people who have aspiration problems, like alcoholics or the elderly, as well as Yersinia pestis, Lawton's model organism. Pseudomonas aeruginosa, causative agent of folliculitis, amongst other things. And Bordetella pertussis, or the causative agent of whooping cough. All of these and many other bacteria use the chaperone-Usher pathway to facilitate their infection. So what we understand about the chaperone-Usher pathway using our E. coli model system may likely be generalizable to many, many different bacteria. As I mentioned, UPAC is our model system. It uses different chaperone-Usher pathways, termed either the FIM pathway or the PAP pathway, to secrete pili onto its surface, where these pili can adhere to the urinary tract and cause UTI. This is an electron micrograph of an E. coli cell, and you can see that this E. coli cell is decorated by these hair-like projections or pili. Here's an overview of the assembly pathway of pili, the chaperone-Usher pathway. So this is a mature pilus seen here. It projects out of the bacteria's cell to in a place where it can adhere to human tissue and act at a distance. So I'll orient you briefly here. This is the extracellular milieu on the top of the screen. On the bottom of the screen is the bacterial cell. And bacterial cells are encapsulated by two membranes, an outer membrane and an inner membrane. And in between these two membranes is what's termed a periplasmic space here. So the chaperone-Usher pathway commences when pilus subunits are translocated through the inner membrane through the sec translocon. Upon gaining access to the periplasm, they're bound by a partner chaperone protein pictured here in yellow, and the chaperone facilitates their folding and aids in their targeting to the outer membrane, and particular to this outer membrane Usher molecule. The Usher has 5 different domains, and I'll introduce them shortly. But the job of the Usher is to catalyze the exchange of a chaperone-subunit interaction in the periplasm for a subunit-subunit interaction in the mature pilus. So this process continues in a sequential manner, and the pilus grows, starting with its adhesive tip and working its way down. And I'll show that in a moment in an animated form that I think brings this pathway together. All of this happens in the absence of any external energy source, so there's no ATP, no proton motive force present in the periplasm. All of this occurs and is driven in the forward direction due to the way that these protein-protein interactions occur. So a chaperone-subunit complex protein-protein interaction exists at a much higher energy state than a subunit-subunit interaction in the mature pilus. So this is an energetically favorable process, doesn't require ATP. I'm going to zoom in on this Usher now. We're very lucky we have some crystal structures of the Usher available. As I mentioned, it's a 5-domain protein. It contains this outer membrane channel, the barrel domain, and 4 additional periplasmic domains, an Ndomain, 2 C domains, and a plug domain. And I want to draw your attention to this global conformational change of the Usher. So when the Usher is in an inactive state, it's held occluded by its plug domain. The plug occludes the barrel lumen, and it prevents influx and efflux to and from the bacterial cell. As you can see in this structure, the other domains had to be cleaved off to solve this X-ray crystal structure, so we don't know where they exist when the Usher is inactive. But upon some activation trigger, the Usher undergoes this global conformational change. The plug becomes expelled from the barrel lumen and then can lie in the periplasm adjacent to the Usher's endomain. And this is a top-down image of the Usher. You can see the plug including the barrel lumen, and upon activation, plug is expelled from the barrel lumen, and the lumen also becomes much rounder so that it can accommodate a growing pilus. And this is an image of this Usher in the active secreting a pilus. This is the first pilus subunit bound by its chaperone. So I'll show this pilus assembly mechanism in an animated form now. This is the Usher. Barrel is inserted in the outer membrane. Plug is occluding the barrel lumen because it's inactive. The Usher first recruits a chaperone-subunit complex. This is the chaperone adhesion complex, the first one to target the Usher. And the Usher undergoes some trigger event in which this plug is expelled from the barrel lumen, and the handoff occurs. So the chaperone-subunit complex is handed over from the endomain to the C domains of the Usher. And this is a critical event in that it frees the endomain for the recruitment of the next chaperone-subunit complex. The Usher orients these subunits perfectly relative to one another so that they can participate in this donor strand exchange event in which a chaperone-subunit complex interaction is outcompeted for subunit interaction. And this handoff from N to C domain continues, and the process continues in the cyclical manner as the pilus is assembled, starting with its adhesive tip and working its way down to the pilus rod. This is similar to the mechanism that I've just shown you. It highlights that handoff event, though. Here's the Usher bound by its first chaperone subunit complex. Its endomain is free to recruit the next chaperone subunit complex, and a handoff event occurs from the N to the C domains. Now we know something about this handoff event. We know that it's the same interface of the chaperone molecule that binds the endomain in the Usher's first binding site as the interface of the chaperone that binds the Usher's C domains after that handoff occurs, suggesting some sort of competition of the Usher's two domains for the same binding site on the chaperone. So the first question that I asked is, how does the Usher recruit these chaperone subunit complexes to its endomain? And then how does the Usher hand these off from the endomain to the C domains? And we go about this to try to understand potential targets for novel small molecules. For example, can we prevent recruitment to the Usher? Can we prevent handoff from the N to the C domain? So we want to understand this mechanism with that in mind. So to do this, we used a single site labeling technique. I specifically labeled the chaperone subunit complexes with a fluorophore. And the important property of this fluorophore is that it increases in fluorescence as it's bound by a partner protein. So as I titrate in one protein to the other, the fluorophore will increase in fluorescence in such a way that I can measure it and determine exactly how strong this binding interaction occurs. So I wanted to introduce this label at a very specific site on the chaperone, a site in which the label interacts closely with the Usher's endomain in its first step. And this is shown here. This is the Usher's endomain, chaperone with its labeling sites in red, bound to the pilus subunit. So these interact in very close proximity to the Usher's endomain, and the same is true of these labeling sites at the Usher's C domain. So these labeling sites serve as a nice assay for us to measure the chaperone subunit complexes binding to the Usher's first site and second binding sites. So we performed a series of experiments. The first thing that we did was monitor the interaction of the full-length Usher for chaperone subunit complex. As you can see, this binds at a very high affinity. So what I'm graphing here is the KD. KD is inversely proportional to affinity. So the lower the KD on the graph, the stronger the binding interaction. And then I deleted the C domains in succession of the Usher. I deleted the C2 domain and then the C1 and C2 domains together. And as you can see, there's a major drop-off in this affinity here, suggesting that these C domains provide the Usher's high affinity binding site. And these data also beg the question that if these C domains are the high affinity binding site of the Usher, then why do chaperone subunit complexes not bind directly to the C domains? Why do they first bind to the N domain? And we believe that that was because the C domains were held inaccessible in some way, or maybe masked in the absence of endomain binding. So to get at that question, we deleted the Usher's endomain, and we no longer detected any affinity. I checked up to about double the concentration, and we were no longer able to detect any binding, suggesting that in the absence of binding the Usher's first binding site, the C domains are not free to bind chaperone subunit complexes. We hypothesized that these C domains are masked by the Usher's plug domain. So to get at that question, we deleted the plug domain. We saw no difference in overall affinity of the Usher, but we hypothesized that if we delete the plug domain in the absence of that N domain, that we would be able to restore the affinity of the Usher's C domains. That's exactly what we saw. So we restored the affinity from upwards of 1.3 micromolar to nearly wild-type levels, suggesting that the plug domain masks the Usher's C domains in the absence of N domain binding. So taken together, these data provide us with a model in which the Usher's plug domain includes its barrel lumen when it's inactive. We knew that already, but we also now understand that the Usher's plug domain is masking its C domains. So upon recruitment to the Usher of chaperone subunit complexes, the Usher undergoes this activation trigger and the C domains are able to become free. And then these C domains are the high affinity binding sites, so this handoff from N to the C domains is likely driven by differential affinity, so that handoff can occur. And we believe that this phenomenon of plug domain masking may be a way that the Usher can ensure that every pilus it assembles contains the adhesive subunit at its tip, in a place where it can adhere to the human urinary tract and facilitate infection. Now, these results were, these data gave us some very interesting results, helped us elucidate the model for Pilus Biogenesis, but they required us to delete Usher's domains. And it posed the problem that we don't understand how all of these domains interact with one another in the complex of Pilus assembly. So we wanted to study these interactions in the context of the full-length Usher, so we modified our assay a little bit. And in this case, we performed what's termed a FRET-based assay, stands for fluorescence resonance energy transfer. Similarly, we labeled the chaperone of chaperone subunit complexes with one fluorophore, and we labeled the Usher with another fluorophore. And when we choose these fluorophores very carefully, we can monitor the transfer of energy between them. And the transfer of energy between them is a function of how close they are to one another. So as I titrate in one protein to the other, the fluorophores become much closer, they undergo energy transfer, and I can measure that, and from that I can establish exactly how high of an affinity these interactions are. So I want to introduce a couple of the structural differences between the subunits of these pili. The first subunit, the adhesive subunit, termed FimH, contains two different domains. The only subunit to contain two domains. It contains a lectin domain. This is the portion of the molecule that binds to the urinary tract. And it contains a pilin domain, similar to all of the other subunits. So all subunits contain this pilin domain, only the adhesive subunit has the additional lectin domain. So we performed some of these FRET-based assays, and the first thing we did was monitor the interaction of chaperone subunit complex for the full-length Usher. We labeled the N domain of the Usher and the chaperone of chaperone subunit complex, and we detected an affinity of about 200 nanomolar. I want to draw your attention to this affinity because this corroborates our previous single labeling results wherein we deleted the C domains and the only accessible binding site was the N domain. This is a very similar readout, suggesting that our single labeling fluorescence results hold true in the context of the full-length Usher. We hypothesized that we also performed the same experiment in the context of an additional chaperone subunit complex. This is FimCG. It lacks a lectin domain, and we no longer detected any affinity. So we were interested in what is it about this chaperone subunit complex that provides this high affinity binding to the Usher's N domain. So we hypothesized that it was the lectin domain. We hypothesized that the difference between FimG and FimH was that FimH has that additional domain. So we thought that if we delete this domain, we should be able to no longer detect affinity. However, when we deleted the domain, we still detected binding to the Usher's N domain, suggesting that it's not the lectin domain, but rather the difference in the pilin domain between FimH and FimG that provides this high affinity of FimH for the Usher's N domain. So to confirm this, we grafted the lectin domain of FimH onto the pilin domain of FimG, and we were not able to detect any binding. So these data taken together suggest that it's the pilin domain that's important for the recruitment to the Usher. We were also interested in understanding how the Usher becomes activated. So we modified our assay a little bit. In this case, we labeled the Usher with two of our fluorophores, the donor and the acceptor. And same principle, as these fluorophores become closer to one another, they undergo energy transfer. And I can measure this energy transfer. So as I titrate in one protein into the other, I measure how this transfer of energy occurs from one fluorophore to the other. And this provides a readout for Usher activation by measuring plug expulsion. Again, FIMH is the only subunit to contain both of these domains. All of the other subunits only contain a pilin domain. So a very similar series of experiments. The first thing we did was monitor Usher activation in the context of FIMCH, and we detected a very robust plug expulsion in this case. We hypothesized that the capacity of FIMCH to cause this robust Usher activation was due to the lectin domain of FIMCH, and that's exactly what we're able to determine. We deleted the lectin domain, and as you can see, we can no longer detect any Usher activation. Similarly, with an additional chaperone subunit complex, we couldn't detect Usher activation in this case either. So these data together suggest that the lectin domain of FIMH is necessary to activate the Usher. And the question was, is it also sufficient? So we grafted the lectin domain of FIMH onto the pilin domain of FIMG, and we were able to restore activation of the Usher, suggesting that this lectin domain is necessary and sufficient to activate the FIMD Usher. So these data, together with some additional data that I haven't shown you today, suggest this two-step verification system of the Usher by which it identifies and incorporates chaperone subunit complexes. So we know from the literature that the Usher's end domain can bind any chaperone subunit complex. In this case, I've shown it binding first to FIMCG. But because FIMCG lacks that lectin domain, it cannot activate the Usher. So it dissociates from the end domain, and the end domain is again free to recruit an additional chaperone subunit complex. In this case, the FIMH chaperone subunit complex is recruited, and this binding to the end domain is mediated by the pilin domain of FIMH. Because FIMH has that lectin domain, it's able to trigger the activation of the Usher. So the Usher is activated, and then handoff can occur from the Usher's end domain to the C domains, and the process can continue in this cyclical manner as the pilus is assembled, starting with the adhesive tip and working its way down to the pilus rod. So far I've shown you the Usher in its monomeric form, but we know that the Usher actually exists in membranes as an oligomer. So more than one Usher are present together in an Usher complex. However, in all of the structural studies that have been performed, we only determined that one of the Ushers of the complex is actively translocating a pilus, which begs the question, what is the other Usher doing? Is the other Usher involved in pilus assembly at all, or is it just standing by idly? And we hypothesized that the additional Ushers are indeed contributing to Pilus biogenesis, because it would be incredibly energetically expensive for these Ushers to be assembled and secreted into the outer membrane of a bacterium if they had no function. So we performed an experiment, and the idea was that if these Ushers are able to interact with one another, both of these, because they have domains deleted, each is separately inefficient for Pilus biogenesis, but if we add them together, they should be able to provide all of the domains necessary to assemble pili. The idea was that chaperone subunit complex would first be recruited to the end domains of this Usher, and then could be transferred over to the Usher's domain and Usher's C domains to the Usher on the left there. So we performed this experiment, but as you can see in this electron micrograph, we did not detect any pilus formation. And this result left us confounded for a while, but based on our recent fluorescence binding studies, we then understood why this experiment wasn't working for pilus assembly. We understood that in the Usher on the left, the plug domain was masking the C domains. So we hypothesized that if we perform the same experiment, but in this case, in the absence of the plug domain, in the background of the end domain deletion, we should be able to free up these C domains by unmasking them. And then the handoff from N to the C domain should be able to occur, and that the pili biogenesis would be restored. And that's exactly what we were able to determine. Hopefully you can see in this image, these bacteria are decorated with pili on their surface. So these were some of the first data that suggest that these ushers can work together in trans for Pilus biogenesis. So in vivo, FIMD is functional in trans for subunit recruitment, assembly, and secretion, and this provides conclusive evidence for a functional usher oligomer in vivo. And this likely serves to increase the efficiency of assembly. And the requirement to delete this usher's plug domain demonstrates that domain masking keeps the usher in its inactive state in vivo. And we think that this ensures that every pilus that's assembled contains the adhesive subunit at its tip in a position to adhere to the urinary tract and cause infection. So I've made a model here that encompasses all of the data that I've shown you today, as well as some additional data. Here is the usher pictured as a trimer in the outer membrane. As you can see, we have different chaperone subunit complexes in the periplasm. And the first chaperone subunit complex, the adhesin, is recruited to the usher's endomain. And this is the first step of the usher's two-step verification system. It checks that the pilin domain of FIMH is correct. So this initial recruitment is mediated by the pilin domain of FIMH. And the second step of the two-step verification system checks the lectin domain of FIMH. So the pilin domain is like FIMH's password. The lectin domain is like FIMH's street it grew up on, or mother's maiden name, or favorite animal, or something like that. That's the second step of the verification system. So the usher checks for that. The plug is expelled from the barrel lumen. The usher becomes activated, and the handoff can occur. And then the endomain is free to recruit additional chaperone subunit complexes. And the handoff continues. And the process continues as the pilus starts to be assembled, starting with its adhesive tip and ensured to start with its adhesive tip, and works its way down to the pilus rod. And we think that these additional ushers in the oligomer are able to provide additional platforms for the pilus rod subunit, because these rod subunits, over 1,000 copies of these rod subunits are necessary to assemble the mature pilus. So we look at this model with an idea to disrupt aspects of this pilus assembly mechanism. Can we prevent recruitment to the usher? Can we disrupt the usher's oligomeric interface? Can we prevent this affinity differential that leads to the handoff from the N to the C domains? So we now have hopes to start screening libraries of small molecules that can interrupt any of these aspects of the mechanism. And our fluorescence-based assays are well poised to do so. So overall, the conclusions of the data I've shown you today: FIMCH binds the N domain of the usher first because the high affinity C domains are masked by the plug domain. The N to C handoffs driven by differential affinity. The lectin and pilin domains each have a distinct role in the recognition of the usher for subunit incorporation. And the usher is functional as a oligomer, and novel therapeutics may now be designed to target the newly elucidated steps of the chaperone-usher assembly mechanism, and these provide promising avenues for the treatment and prevention of UTIs. And this is a nice sand formation that I found on the beach that, to me at least, looked remarkably like an E. coli cell decorated with pili. And with that, I thank especially my mentor, Dr. Tanasi, and Nadine Henderson and Hemel Chauhan, who contributed directly to this project, as well as other lab members, mentors, administrators, and funding agencies. And I thank the Office of the Provost for the invitation to speak, and I thank all of you for your attention.